Hi everyone, Teacher Janet here. Welcome back to my educational channel on biology. In this video, we'll be discussing application of enzymes in daily life. And this is from subtopic 5.3 of the Biology Form 4 KSSM syllabus. So in this video, we'll be discussing some wonderful and versatile uses of enzymes like protease, cellulase, and lactase in industry and in daily life. So enzymes have benefited mankind in many different ways, right? And very often we use commercial products or consume food products that contain enzymes or need the help of enzymes to be produced. For example, agar-agar, right? So we are going to discuss the uses of enzymes in the food processing industry, leather industry, in the production of biodetergents, also in the production of lactose-free milk here, and so forth. Let's find out more about the uses of enzymes. The learning standards for today's lesson are as follows. 5.3 Application of enzymes in daily life. So after this lesson, we should be able to explain by using examples the application of enzymes in daily life. So we're going to discuss the uses of different types of enzymes in different types of industries or sectors. 5.3 Application of enzymes in daily life. So enzymes are widely used in the commercial sector and at home in daily life. Now these enzymes are extracted from natural resources such as bacteria. So they are produced by using bacteria or fungi, and they can also be produced synthetically. So they can be produced from microorganisms like bacteria on a large scale. Now the use of enzymes in industry involves a technology called immobilized enzyme technology, which we will discuss in the next slide. So you can see it on the picture here on the right. So in immobilized enzyme technology, the enzyme is bound to or combines with some inert substance here, and then it can carry out the catalysis of certain reactions. We'll discuss more in the next slide. Now, apart from discussing the meaning of immobilized enzyme technology, we will also discuss the applications of enzymes in daily life. And there are many applications of enzymes. For example, in the production of lactose-free milk, fruit juice, meat tenderizer, cheese, agar-agar from seaweed, syrup, cereal grain products, and also in the production of leather products like shoes and bags, in the production of bio-detergents for washing clothes, and the production of digestive enzyme pills for those who suffer from indigestion. Let's find out more about these interesting applications. Now let's have a look at this question which may be asked in the exam. Since this is a new topic on immobilized enzyme technology, okay, it's a new part of the syllabus. Explain the use of and advantages of immobilized enzyme technology. 6 marks. So the word immobilized means to prevent something from moving. Now what is prevented from moving? The enzymes. Okay. So how are the enzymes immobilized or prevented from moving? They are combined with inert or inactive and insoluble substances. So I give you an example here. The substance is calcium alginate, but you do not need to write it down for the exam because it's not in the textbook. All right, just for your knowledge. Now, so immobilized enzymes are enzymes that combine with inert and insoluble substances. This will help to increase the resistance of the enzymes to changes in factors like pH and temperature. So we know that enzymes are sensitive to changes in pH and temperature, but when they are immobilized and bound to the inert or active insoluble substance, like in this column here, the immobilized enzyme is attached to the beads or particles of inert substance. This will increase their resistance to changes in factors like pH and temperature so that the reactions can be carried out at maximum rates. All right, so that the enzymes are more effective in catalyzing the reactions. All right, if not, if there are some tiny changes in temperature, then enzymes may be denatured, for example. Okay, so example of immobilized enzymes is the enzyme lactase that is used to digest lactose in milk in order to produce the lactose-free milk for lactose intolerant people. So we'll be discussing that in the next slide, right? Now, so the enzyme lactase can be bound to the bits of inert substance here, okay, in the column here, and then milk is passed through, poured in from the top, right? Then the lactase will digest the lactose in the milk to form simpler substances. Then lactose-free milk will be produced down here, okay, which is suitable to be consumed by lactose intolerant people. We'll discuss more of this in the next slide. Now, the advantages of immobilized enzyme technology. All right, you're asked to give the advantages. So the first part is on the use of immobilized enzymes with one example stated here. So now for advantages. Immobilized enzymes are more resistant to changes in pH and temperature. So this will help them to catalyze reactions at maximum rates because they are not so affected by changes in pH and temperature. Then it lets the enzyme molecules remain in the same position, held in the same position or held in place on the inert substance throughout the catalytic reaction. So after that, the enzymes can be easily separated from the products. Okay, the product will flow out here. The enzymes remain inside the column here. So they can be used again to catalyze further reactions. Now, how is lactose-free milk produced? So lactose-free milk is milk that does not contain lactose. Why do you want to remove the lactose? Because certain people can't drink milk. They don't have the enzyme lactase, or they don't have enough of lactase to digest the lactose in the milk. So this undigested lactose in the milk that is drunk causes diarrhea, stomach pain, and other symptoms in the digestive system of the lactose intolerant people who secrete too little lactase, enzyme lactase, or none at all, right? So therefore, that milk can be produced which contains no lactose to help these people be able to consume milk. All right, because milk has many benefits, such as a high calcium content. Okay, so let's see how it's done. Now, in the production of lactose-free milk, the milk that contains lactose, which is a disaccharide, okay, that looks like that, two molecules of monosaccharides attached together. So this milk that contains lactose is passed over the immobilized enzyme lactase that's attached to the bits of inert substance, right? So this is the enzyme lactase in the bits. Yeah, the enzyme lactase is combined with or bound to the bits of inert substance. As the milk flows through the immobilized enzyme, the enzyme lactase on the surface of the bits will hydrolyze the lactose in the milk to form glucose and galactose. Okay, so now the lactose has been converted to monosaccharides. And these two substances do not cause any reactions in the lactose intolerant people. In fact, it is easily absorbed by the lactose intolerant people. Now, so D is the production of the lactose-free milk. Now, so people who are lactose intolerant can drink this lactose-free milk without any fear of getting diarrhea or stomach pain. All right. So here are the notes for the production of lactose-free milk. So we have discussed the diagram just now, and let's go through these notes very quickly. Milk which contains lactose is poured into a tank with a column that contains immobilized lactase, which is the enzyme. So this immobilized lactase is attached to the inert substance inside the column. Then the enzyme lactase hydrolyzes the lactose into glucose and galactose in milk. So this is the equation. Lactose plus water with the action of lactase. Lactose breaks down to form glucose and galactose. Thus, lactase removes lactose from milk to produce lactose-free milk. And this milk is suitable for people who have a lack of the enzyme lactase in their digestive system. Okay, so actually lactase is produced by the small intestine. And these people have a lack of this enzyme in the small intestine. So they suffer from lactose intolerance. So intolerance here means that the people who have a lack of lactase cannot tolerate or bear the presence of lactose, right? So they will have difficulty digesting the lactose and they will show symptoms which are stomach pain. Nausea means the feeling of wanting to vomit, and then diarrhea, okay, or chirit-birit. So sometimes, there are people who drink milk and then after that they get the stomach ache or they have to run to the toilet because they have diarrhea. So they think they are allergic to milk. Actually, they could be lactose intolerant. That means it could be due to the fact that they have a lack of this enzyme lactase, right? That causes them to be unable to digest the lactose and so have all these symptoms. All right. So one way to find out is to consult the doctor about the symptoms. And sometimes if we stop taking the milk and we find that the stomach pain goes away or diarrhea goes away, so it is a sign that maybe the person has lactose intolerance. All right, but can go to the doctor for a consultation, okay, on this. So for people like this, they have to drink the milk that is lactose free, which is produced by using the immobilized enzyme lactase. Okay, so lactose free milk, normally when you buy the milk, can check the label on the milk bottle or the milk carton, and if it is lactose free, it will be stated there lactose free. Okay, so here are some short notes whereby we will discuss various types of enzymes other than lactase that are used in industry, okay, to produce various products. So for the table, there will be the titles industry, then name of enzyme, use of enzyme, and the equation for the enzymatic reaction. Because sometimes in questions, they will ask you, state an example of the use of enzyme in, say, food industry or food processing industry, right? So you have to think of all the different types of enzymes used in food processing. Or it could be the use of enzyme in production of leather or something else, right? So it's good to have these titles. So the first 8 enzymes are used in food processing, all right? Now, number 1 is lactase, as we have discussed, which is used to produce the lactose-free milk. So here is the short explanation for it. Lactase hydrolyzes lactose in milk into glucose and galactose to remove the lactose from milk. And this is suitable to be drunk by lactose intolerant people. And then the equation, as we have mentioned, is lactose plus water, arrow, and then write the name of the enzyme on top of the arrow, produces glucose plus galactose. All right, so let's look at the other enzymes that are involved in food processing and also other industries. Right, the second product that is produced with the help of enzymes are the fruit juices, like the clear apple juices or clear apple juice or orange juice and so forth. So the type of enzymes used to produce these fruit juices are pectinase and cellulase. These are the two enzymes used. Now, what is pectinase? Pectinase is an enzyme that breaks down pectin in the cell walls. So what is pectin? Pectin is a jelly-like substance in cell walls that helps to cement and bind the plant cells together. It's in the cell walls and between the cell walls, right? So the pectinase breaks down the pectin in the cell walls through this equation, pectin plus water. Pectinase should be written on top here, right? Produces the monomers, the monosaccharides. So when the pectin is broken down, it will cause the cell walls to separate from each other. It can also break down the cell walls. Now, apart from that, there's also cellulase, which helps to break down the cellulose that is found abundantly in cell walls. Okay, so in this way, these two enzymes help to break down the cell walls of the fruit cells so that more juices can be released from the fruit cells. Okay, apart from just mashing up the fruit, if you want to really release the juice from the fruit cells, we have to break open the fruit cells, right? So these two enzymes will digest the cell walls, break down the cell walls, and release the juices from many fruit cells. So this will increase the quantity of fruit juice obtained and reduce the cloudiness of the fruit juice. How does it reduce the cloudiness of the fruit juice? Because the pectinase will hydrolyze or break down the insoluble compounds that are suspended in the fruit juice. All right, and through that way, the fruit juice will become clear, clarified. This is called the clarification of the fruit juice. So it won't have all the bits and pieces floating on top or inside the juice, like what we naturally get when we blend some fruit juice, right? Okay, so here in this last column, we see the action of pectinase, which we have discussed, and now the action of cellulase is like this. Cellulase hydrolyzes cellulose with the help of water to produce glucose. So glucose is a soluble substance, right? That is sweet. So it will be found, it will be produced, and then it will be inside the fruit juice. Thirdly, in meat, the processing of meat, especially for housewives. So many types of meat are quite tough, and when the housewife has cut the meat into cubes, right, and needs to maybe fry it, she can use an enzyme called protease to tenderize the meat by digesting the protein in the meat to form soluble polypeptides. So the connective tissue in the meat will be digested to form soluble polypeptides, and in this way the meat will be easier to tear apart, okay, to break and to cut and to tear apart. So usually before cooking, the meat will be sprinkled with the protease. Now, where to get protease? Protease can be obtained as in the supermarket as a meat tenderizer. Okay, it's labeled as meat tenderizer. It comes in a small bottle, and it is meat, and it has a whitish substance. Okay, the meat tenderizer is actually a whitish powder, okay, which can be sprinkled on the meat during the process of marination of the meat. When the meat is mixed with other ingredients and then left for a while, maybe for half an hour or so, for all the ingredients to be absorbed into the meat. So the tenderizer can be put then to tenderize the meat for a while before stir frying or cooking. Now, fish. In the preparation of fish, for example, canned salmon. Salmon, for example, has a tough skin, all right, thick and tough skin that encloses the meat. So in the preparation of canned salmon, this fish skin is separated from the fish meat by using protease, which will dissolve the connective tissues on the skin, and then the skin can be easily separated from the fish meat before canning the fish. So the equation is the same. The protease digests the protein, hydrolyzes it to form soluble polypeptides. Now here's an SPM HOTS question, which has come up before. A housewife wraps some meat in papaya leaves for 3 hours before cooking it. Explain the reasons for action. Okay, 4 marks. So this question is also found, something similar to is found in your textbook. Okay, on this page, it is in the summative practice for this chapter, chapter 5. So underline the keywords. Meat wrapped in what? Papaya leaves, 3 hours. All right. So the papaya leaves contain an enzyme called papain, which is a protease. That means it is an enzyme that digests protein. All right. Digest the meat into simpler compounds such as soluble polypeptides. So in this way, the meat will be tenderized because the connective tissues will be digested, and the meat will be softer. When we cook it, after cooking it and eat, when you eat it, the meat is very soft and easy to cut and chew. Now, another thing that we can explain is why is it wrapped for 3 hours here. So it is to ensure that the enzymes have time to gradually break down the protein in the meat, because enzymes cannot just suddenly break down the protein. They need time to work effectively, all right, to digest the protein in the meat. It is a type of digestion. So 1 mark for each statement here. Next, in the production of syrup, the enzyme amylase is used to hydrolyze the starch to form sugars to make syrup, which is a sweet sugary liquid that we can use to sweeten drinks like fruit juices. So the reaction is as follows. Starch plus water, through the action of amylase, the enzyme amylase, maltose is formed. So maltose is the sugar form that is sweet. It is a disaccharide that is a complex sugar and it is sweet. So it can be used to make this syrup instead of using the normal sugars like cane sugar, which is sucrose. Next, in the formation or production of dairy products such as cheese, lipase can be used in the ripening of cheese. So what's the ripening of cheese? It is one stage in the processing of cheese where the cheese will mature and develop its own texture and flavor. So various enzymes are used to break down substances inside the cheese. For example, lipase is used to break down the lipids in the cheese through this reaction. Lipids plus water, with the action of lipase, produces fatty acids and glycerol. Now, how about renin? Renin is another enzyme that is used to solidify milk proteins to form cheese. So this is the beginning stage of the formation of cheese, where milk has to solidify, curdle and solidify to form the substance that will later form cheese. So renin is added into the milk so that it will coagulate and form cheese, right? To form a substance that is processed to form cheese. So what happens in the coagulation and of the milk proteins? Actually, the renin added will convert the soluble casinogen, the protein called casinogen, into insoluble casein in the milk. So the milk, there'll be clumps of solids in the milk after you put in the renin. All right, and then these solids are used to form the cheese. Cereal grain products. In the production of cereal grain products, the enzyme cellulase is used to break down cellulose to remove seed coat from the cereal grains, right? So in the production of cereals, now some of us eat cereals in the morning as for breakfast. So cereals are actually grains that are eaten as food. For example, they may be maize, wheat, rice, and other cereals like barley. Barley is also a cereal. So if you look at the wheat, right, the seed of the wheat plant has a tough seed coat, a purple layer here, that needs to be broken down when we are producing the cereals. All right, so cellulase is added to break down this tough seed coat because the cellulase can digest the cellulose in the seed coat to form glucose. That way the seed coat is removed in the processing of the cereals to make the cereal products. Now, lastly, for food products, there's the seaweed products like jelly, agar-agar, okay, or jelly powder. So we need the enzyme cellulase to break down the cell walls of seaweeds to release and then extract the agar-agar from the cells. In the making of the agar-agar powder, which looks like a whitish powder that's used to make delicious jelly to be eaten. So how is the jelly made? Now, the seaweeds that are found in the sea, right? For example, usually it's a reddish type of seaweed that's used. They have a lot of this agar-agar in the cells, but it needs to be released. So cellulase is added again to act on the cell walls and digest the cell walls so that the agar-agar can be released. Then after that, the agar-agar is extracted and isolated to make the agar-agar powder, which after that we can use to, when we cook it and add sugars and coloring, it will form the jelly that's very delicious to eat. Next, in the production of leather products. So trypsin is used to extract fur from animal hide or skin in the making of leather products, such as leather shoes, belts, wallets, even leather furniture that has leather chairs, and so forth, right? So leather is actually a strong and flexible material obtained from the chemical treatment of skins of animals such as cattle, sheep, goats, horses, seals, and alligators. So it's the skin, the animal skin, but the animal skin sometimes contains fur or hair. So this trypsin is added to digest the fur or hair, which is actually a type of protein, right? So the hair will come off easily after the trypsin is put on the leather. And the equation for the action of trypsin is polypeptides is digested to form peptides. Next, in production of biodetergents. So nowadays, our detergents contain different types of enzymes that can be used to digest different types of stains, right? Sometimes the detergent is in a liquid form, or sometimes it's in a powder form, all right? So when we wash the clothes that are stained in water containing these detergents, then the enzymes will act on the stains and digest them so that the stains can easily come out. Now let's see what are the enzymes that are found in the biodetergents. First one is cellulase, which dissolves the cellulose stains from clothes, especially from food, right, that have a lot of cellulose on it. So for example, vegetables, cooked vegetables, and fruits, they may cause some stains on the clothes, and these stains may contain cellulose. So cellulase will digest the cellulose to form glucose, which is soluble. So the stain can be easily washed out from the clothes. Now, lipase digests grease or oil stains from clothes. So if we have any greasy stains on the clothes, or even grease from that is from the that's on the cars and vehicles, all right, after repair, maybe some grease has stained the clothes, right, after repairing cars or vehicles, or even for housewives that or people who eat oily food, okay, they may have all these oil stains on the clothes. And the lipase can digest the oil, which is a lipid, to form fatty acids and glycerol. Okay, now when you write equations, remember to write the enzyme above the arrow. All right. Now, normally they don't ask you to write equations, but here I'm writing the equation in case it comes out as a HOTS question. And also, most of these equations will be studied when you study digestion of food later on in chapter 9. All right. So here is for your understanding, to help you to understand how the enzyme works. Thirdly, the enzyme amylase removes starch stains from clothes, such as stains that contain gravy, okay, gravy from cooked food. And lastly, protease dissolves protein stains and also blood stains. Maybe there's some bleeding, some wound on the body, and it stained the clothes. So blood stains contain proteins, which actually comes from the hemoglobin. Hemoglobin in red blood cells contain proteins. It's a type of protein. So the protease can break down the protein stains or blood stains. According to this formula, protein plus water produces polypeptides, okay, when the protein is acted on by the protease. So these polypeptides are again soluble in waters, and they can be washed off easily. So these enzymes will make the detergents very effective in that once we soak the clothes in water for a while, the enzymes will get to work, react, and digest these stains or hydrolyze the stains. After that, the housewife just needs to scrub a bit, and the clothes will be clean. Number 11, for pharmaceutical products and medical products, we have digestive enzymes in the form of pills or tablets. So these enzymes are, for example, lipase, amylase, and protease, which are usually produced by the human body, but in some cases, some people may have a lack of these enzymes in their body. So they can consult a doctor first to find out whether they can eat these pills which contain the digestive enzymes. All right. So these digestive enzymes helps the body to digest or break down food and increase nutrient absorption. All right. For example, the lipids are broken down to form fatty acids and glycerol, which can then be absorbed into the wall of the intestine, into the body. Starch is hydrolyzed by amylase to form maltose, and protein is hydrolyzed by protease, for example, pepsin, to produce polypeptides. So this can help to treat indigestion, gas, and nausea in certain people who have a lack of these enzymes in their digestive system. And also in people with damaged digestive glands, for example, if the pancreas is damaged, right, or taken out, then pancreas actually secretes 3 enzymes, lipase, amylase, and trypsin. So these enzymes can be eaten to replace that which is not produced anymore by the pancreas, right? So that they can continue to digest the various complex food substances. Now, gall bladder is an organ just under the liver here. Its job is to produce a chemical substance called bile, which helps to break down the big lumps of lipids into small droplets for faster enzymatic reaction. So without the gall bladder, the lipid digestion is a bit slower. Thus, lipase can be given so as to speed up the digestion of lipids in the small intestine. All right. Right, let's look at further questions. Some of these are SPM questions, right? Now, number 1, explain how enzymes are used to extract agar-agar from seaweed. Okay, this is the past year question, and the marks are 2 marks. So, secondly, state a role of the enzyme lipase in the food industry. So, highlight the keywords like agar-agar from seaweed, enzyme lipase. Let's answer the first question. Explain how enzymes are used to extract agar-agar from seaweed. So we have to know the name of the enzyme, right? What's the name of the enzyme? Cellulase, right? So the enzyme cellulase breaks down the cell walls of seaweed by hydrolyzing the cellulose, right? And then so that the agar-agar can be easily extracted from the cells. Okay, so if it's 2 marks, you can write like this, or if it's 3 marks, you can also write the action of cellulase, which is to break down the cellulose to form glucose. So in that way, the cell walls will be broken down. Now, for lipase, state the role of lipase in the food industry, just 1 mark. So lipase is used in the production or ripening of cheese. And if there are more marks, you can write, it helps to digest the lipids in the cheese to form fatty acids and glycerol. Okay, right. The last question here is a hot question. Explain why enzymes from bacteria living in hot springs are suitable to be used in the biodetergents for washing laundry or clothes. Why do we use enzymes from bacteria living in hot springs? Okay, so the reason is because in the hot springs the bacteria has enzymes which are resistant to high temperatures, right? They are not destroyed or denatured. Okay, let's look at the answers. So 4 marks here. Firstly, we can mention that in washing machines, there's a function for temperature control. All right, your washing machine at home may have this function where you can increase the temperature from room temperature to higher temperatures like 40 degrees, 50 degrees Celsius, 60 degrees Celsius, and so forth. Now, if the machine is set at high temperatures, what will happen to normal enzymes? We know that normal enzymes like lipase and amylase that are similar to the ones in the human body have an optimum temperature of about 37 degrees Celsius only. So if the machine is set to a higher temperature like 50 or 60 degrees Celsius, the normal enzymes in the detergents like lipase and amylase may denature, meaning that they may be destroyed and they are not able to catalyze any more reactions to remove the stains from the clothes. Okay, so they will not be effective to remove stains from the clothes. Thus, if the enzymes are extracted from bacteria that live in the hot springs, they are able to withstand high temperatures as they come from the bacteria that live in the hot springs. So they will not undergo denaturation when the machine is set at higher temperatures like 60 degrees Celsius. Okay, for example. So some students have asked for the notes related to these slides. So here I have the notes for the table showing the industry, name of enzyme, use of enzyme, and equation for enzymatic reaction, which we have discussed in the previous slides. So here I put it in the table format, so it's easy for you to copy. However, the earlier notes on immobilized enzyme you will have to get from the slides just now, okay, in the earlier section of the video. Okay, so these notes are only for the table, but they will show you the enzymes that you need to know for the exam because the textbook did not list down all the enzymes that you need to know. So here are some of the enzymes that are commonly discussed for this topic, right? So I will not go through all this again, but you can, because we have discussed it in the slides earlier, so you can just pause and then jot down the notes if you want to have it. Right, so this is the second part of the notes, and these notes have been produced very systematically and with a great dedication and work to make sure that it has all the different parts. Normally the equations are not asked, but it will help you to understand the process, and if you have to elaborate in the answers, then you can explain these the actions of the different types of enzymes. All right, apart from explaining the action in words like this, you can also write down the reactions or explain the reactions in words. So you can pause the video and then copy the notes. And there's one more part, the last part here. Okay, now this is the last part of the table, the notes and the table for the types of enzymes that we need to know, some of the enzymes that we need to know. Okay, so that's all for this lesson. So thanks for viewing, and please do share, like, and subscribe. Goodbye for now.